Okay, well, I guess we'll get started this afternoon. Welcome, everybody, to our CCOVI lecture series for this Monday, May 17th. It's my pleasure to introduce my colleague and longtime friend, Dr. George van der Merwe, who's our guest speaker today. Just want to give you a little bit of background on George. He received his PhD from the University of Stellenbosch, but he's actually had a very long association with us here in CCOVI that I'm going to share with you. He did his PhD under the supervision of Dr. Henny van Vuren. He started off his studies in Stellenbosch, but the lab actually moved here to come to the Collaborators and Viticulture Institute. And George actually started here in 1998. He actually also did a year under the supervision of Dr. Terence Cooper during the course of his PhD. Was here for a few years, and then the lab moved to University of British Columbia, to the Wine Research Center at UBC. And so he finished up his studies at UBC and continued there for a few years afterwards, where he really got his interest in further developing systems biology of yeast and applying that to fermentation science. From that point, he actually started at the University of Guelph as an assistant professor in the Department of Molecular and Cellular Biology, and is currently an associate professor at the University of Guelph and also a CCOVI fellow. He officially joined the CCOVI team as a fellow last year and is part of our network of researchers involved in the grape and wine industry across Canada. George is certainly committed to the wine industries here in Canada as well as in South Africa, and he's actually just shared with me he's going to go back and do a sabbatical in South Africa early next year. So with that, it's my pleasure to introduce George's topic today, understanding the impact of fermentation stresses on yeast during vinification.
Thank you, Debbie. Yeah, it's always nice to come back to CCOVI and actually see the familiar faces again and be able to share some time with you guys. It conjures up nice memories to see the Inniskillen Hall again. So yeah, the topic of my research that we have in the lab is to understand how yeast respond to fermentation stresses during the vinification process. So we what we try to do what winemakers try to do in the industry is to actually take the hard-earned grapes that the growers give us and then do some kind of fermentation with it, either by inoculating these fermentations with commercially produced yeast or with spontaneous fermentations. Now, most winemakers actually go through the process of inoculation, and this is the kind of stuff that they see in their wineries, in their refrigerators, where they have these commercially active dry yeast for specific winemaking purposes. And we see these as little balls of clay, they look like almost, that gets rehydrated and then inoculated into the fermentation. However, it's seen as an ingredient by most people. But what we actually have to understand is that these things are microorganisms that live and have a very specific lifestyle that it follows, and consequently it's impacted by the fermentation per se. So our demands to actually make wine doesn't really have a lot of bearing on what the yeast wants to do. So if we look at a simple yeast, what do yeast actually really do? For us to understand how it responds to fermentation stresses, we need to understand that a little bit better. So if we can imagine this would be a Saccharomyces cerevisiae yeast that does most of the fermentations in the wine industry, and it has a plasma membrane which is impermeable to a variety of different solutes in the environment. So most of the ions or the nutrients that are in the environment cannot freely pass across this plasma membrane. And consequently, the yeast has an intercellular environment that it can control very specifically. So it needs to do that very specifically so that it can adapt to a variety of different conditions and also perform the functions that it needs to within the cell itself. Because it has this impermeable membrane, it needs a variety of different proteins, such as transporter proteins, that will actually help to uptake nutrients that are in the environment, to uptake it into the cell itself. Just because these nutrients can't freely pass across the plasma membrane, these nutrients then serve as substrates for a variety of different enzymes, and they produce products that either help the yeast grow, for example, in that it produces biomass that help the yeast to divide and form a new daughter cell, or it produces metabolites, and the metabolites that we know in the industry would be ethanol or flavor compounds. And so these are all products of an enzymatic reaction. So that is typically what the yeast would like to do. This is supported by obviously DNA or genes that are found on the DNA of this organism. These genes are expressed to form RNA. These RNAs are translated into proteins, and the proteins are the workhorses of the cell. They perform all the functions within the cell itself. So these proteins can be transporters, they can be enzymes, or they can even be the biomass. So this is basically what the yeast does, how it would go ahead and grow. So that's very simplistic. If we now go and actually look at the optimal growth conditions of yeast, it has a temperature range of about 25 to 30 degrees Celsius where it likes to grow. It likes to grow in a little bit of a pH of a lower pH and a little bit of an acidic pH at around 5. It doesn't really like the presence of ethanol. So if the yeast cell grows, for example, in nature, on the surface of a berry, it'll produce ethanol, and most of that ethanol would evaporate, or can be caught up in solution and can be used as a carbon source later on. But it doesn't really like ethanol. It prefers certain kinds of nitrogen as well. Amino acids are known to serve as nitrogen sources for yeast. However, it discriminates between the qualities of these different nitrogen sources. So some of them are very good nitrogen sources, like glutamine, for example. And some of them are really poor nitrogen sources like proline or serine. So what distinguishes a good from a poor nitrogen source is just the ability to actually assimilate these nitrogen sources into the cell itself. So ammonia and glutamine are really good nitrogen sources for the yeast, and it prefers that. It likes to grow in the presence of oxygen, and the reason for that is so that it can easily make membrane components, and it likes growing in an environment that has a high water activity, such as a liquid environment, for example. So these are the things that yeast like to do. Now let's shift gears a little bit and look at the constituents of grape juice. We see that there's a high concentration of sugars in grape juice. This is just of a regular table must, so we're not even talking about ice wine here. So we see, say, about between 16 and 26% sugars or degrees Brix. Often we see in the range of about 22% coming into the wineries. There's a high concentration of acids, organic acids, that's part of grape juice. That pushes the pH down. And then there are a variety of minerals that are present in trace amounts. And if we look at the nitrogen levels, they are variable. We can't put a very specific value on this. Some grape juices that come into a winery would have more nitrogen associated with it, while others would be poor in nitrogen. So this varies considerably of the grape juices that actually come in. So what are we asking of yeast then? If we look at a typical wine fermentation, we start with a juice of approximately 22 degrees Brix, and we now inoculate these yeasts into this specific environment. Consequently, these Brix would be used, and as it's being used, the cell mass within the fermentation of the yeast itself would increase. When it gets to a certain concentration of ethanol produced, the yeast goes into stationary phase and consequently does not divide anymore. It doesn't grow. It doesn't mean it's not metabolically active. It is still metabolically active, and it goes and converts most of these sugars into ethanol. And you can see an increase in the concentration of ethanol up to above 12% volume per volume, as you can see over here. So there's an enormous amount of ethanol that's produced during this fermentation. So if we look at this in a little bit more detail, at the starting point when we inoculate the yeast into this environment, there's a high solute concentration, a high concentration of sugars, for example, and other soluble matter in the fermentation itself. That pushes the osmotic pressure in the fermentation up, and the yeast is submitted to this osmotic stress. Right off the bat, the yeast starts with a low pH that it has to combat. That as the fermentation starts, it has to overcome all these organic acids. As it continues, it produces a high concentration of ethanol. that it has to combat. And as the fermentation proceeds, as this glucose becomes limiting, it also uses other nutrient components such as vitamins and also nitrogen sources that also become depleted during the fermentation process. So these are a variety of challenges that the yeast actually has to face. So if we now go and actually compare vinification to the optimal growth conditions of a yeast, then there are a few challenges that jump out. Firstly, most of the fermentations that are done in the industry are not done in the optimal growth temperature. Most of the white wines that are produced these days start with actually a cold fermentation that starts anywhere in the range from 18 down to 14 degrees Celsius. So there's already a temperature challenge as we start off with. The second thing is the pH of the juices are typically below 3.4, specifically in the environment like Canada, like Ontario, where there are huge issues with cold stress and with not enough photon hours to actually get grapes to mature. Often this pH level is closer to 3 and sometimes even below. So there are major challenges for the yeast in that sense. At the end of the fermentation, the yeast can produce up to 16% volume per volume ethanol. This is the normal range is normally between 11 and 12% for Ontario, but that is significantly more than what the yeast likes to encounter. So there's a stress in that part. Nitrogen can become depleted during a fermentation. This is a variable. However, there aren't high concentrations of ammonia and glutamine present within the fermentation itself. There are typically other amino acids that will stand in as a nitrogen source. Arginine would be one of them that would supplement as a nitrogen source. Most fermentations run through an anaerobic environment. In other words, there's not dissolved oxygen around at high levels, and the yeast does not really like this because it cannot produce membrane in anaerobic conditions. So that is a detractor as well. And if we look at the water activity that are present within normal wine juices and ice wine juice, in a normal table wine juice, we run it at a water activity of about 0.982, and in ice wine it can be as low as 0.939. So these are levels that are significantly lower than what the yeast likes to encounter, and this imparts osmotic stress on the yeast itself. So if we look at all these stresses and we add them all up, what the yeast has to do now is to respond to all these stresses and actually make a nice wine for us. That's our demand. The yeast doesn't really care about our demand. What it wants to do is it wants to grow, it wants to protect itself and survive the environment. That's what it focuses on. Our job is to try and find the yeast or manipulate the environment of the yeast in such a way that it ends up making the nice wine that we want. So for us to actually understand how the yeast does this, we have to understand how it responds to all these different stresses. And then once we understand these responses at a molecular level, we can devise strategies to overcome these stresses. Most of these stresses are obviously at a molecular level, so it changes the physiology and the metabolic adaptation of the yeast during this process, and consequently at the end of the day it changes the quality of the wine. I am not going to talk about osmotic stress because we've got Debbie as an expert here, so she's more than likely spoken to you guys about that before, so I'm not going to cover that. I am going to pay a little bit of attention to ethanol toxicity and to nutrient deprivation before we come to my research. So if we look at how the yeast cell functions in a little bit more detail, and we can imagine this would be a plasma membrane, which is a lipid bilayer, as most of you know. Extracellular environment would be here, and this would be the cytoplasm or the intercellular environment. We said in the beginning that it's important for the yeast to control this environment very specifically. So it has a high concentration of potassium ions that it sequesters within the cell to actually allow the cell to be viable and to operate efficiently. It also has a certain concentration of protons to keep the pH of the environment roughly or just below neutrality within the cytoplasm itself. If we now consider the extracellular environment, for example, in grape juice, there's a high concentration of protons because it's acidic, right? So we have a high concentration of these protons. So there's a gradient across the plasma membrane here of high concentration of protons outside and a low concentration of protons inside. Because this is an impermeable barrier, it cannot transport nutrients like amino acids or move nutrients like amino acids freely across this plasma membrane. There are proteins that does this, like amino acid transporters, and these proteins actually transport amino acids into the cytoplasm by also co-transporting protons. So these are symporters. So they bring these protons in, down an electrical chemical gradient from high concentration to low concentration, and consequently also bring amino acids in. So this is how the cell actually takes up nutrients. Right? That's a normal function. What happens if we induce or introduce ethanol? Ethanol can insert within the membrane itself and consequently disrupt the structure of this membrane. It's not as nicely organized as we see over here in the absence of ethanol. So as soon as you move ethanol into or expose the cell to high concentration of ethanol, the fluidity or the structure of this membrane is disrupted. It loses its impermeability status, and consequently some of these potassium ions can leak out. This is a significant stress for the yeast to actually deal with. Also, this electrochemical gradient is disrupted because protons can now move across the plasma membrane as well. In addition to that, ethanol can also disrupt the structure of proteins. It has a denaturing quality to it. So not only proteins within the plasma membrane, such as these amino acid transporters, but also proteins such as enzymes that function within the cell itself can be denatured and consequently can be unfolded. And as soon as this takes place, the ability of those proteins to perform their functions actually decrease significantly. So due to this loss of electrochemical gradient and also the denaturing structure, denaturing of this amino acid transporter, for example, the ability of yeast to actually take up nutrients in a high concentration of ethanol decreases rapidly. So the ability of these yeast to perform fermentations become harder and harder and harder as the fermentation process proceeds and as the concentration of ethanol actually increases. But we still see that it actually can perform fermentation at the end. So what does the yeast do to fight all these different problems that it has to experience? So the first thing that it does is it tries to actually alter the membrane composition. So we know that the plasma membrane is made up of phospholipids and of different kinds of sterols. So what the yeast tries to do is to actually change these phospholipids and increase the production of sterol, specifically ergosterol, to actually try and rescue that disrupted structure of the membrane itself. Problem with this is, for the cell to do this, it needs oxygen. And as we know, in the presence of fermentations, there aren't a lot of dissolved oxygen around to actually support this metabolic function. This becomes a very big stressor during fermentation, as the fermentation proceeds. As the pH of the cytoplasm drops due to the fact that the membrane structure is disrupted, the yeast tries to actually rescue this phenotype by pumping protons out of the cell. So there are proteins within the cell, or that the cell expresses, that actually will pump protons out, and this is in an ATP-dependent manner. So this is a big burden on energy for the yeast to actually cope with this low intracellular pH. The third thing that it does is that the yeast would produce proteins that help with refolding denatured proteins. So we said that ethanol can disrupt the structure of a variety of different proteins. So there are chaperone proteins, and in yeast there are examples like the heat shock proteins that help with actually refolding unfolded proteins or preventing these proteins from these unfolded proteins from forming aggregates within the cell and consequently leading to cell death. So this is a typical response that we see in stresses, in ethanol stress, is this production of heat shock proteins, and we're going to look at this at the end of the presentation in a little bit more detail. What the yeast also does is it produces an intracellular compound, which is actually a complex sugar called trehalose, that function very similarly to chaperones in that it would help with refolding unfolded proteins. We're not going to get into the detail of this in a lot more than that. And then of late, there's been a very interesting observation in the literature in that there are some amino acids that are accumulated at high levels within yeast during ethanol stress. One of these amino acids are proline. Proline is present in abundance within grape juice, but the yeast cannot use it as a carbon source because there's no oxygen around. But what has been found in the literature of late is that proline, for example, can be concentrated at high levels intracellularly and serves as a way to actually combat stress. In bacteria, proline plays a role in combating osmotic stress. And it's thought that this intracellular accumulation of proline within the yeast itself helps with combating ethanol stress. The exact mechanism of how that works is not known right now. So this is what we understand about ethanol toxicity and how the yeast actually responds to this to date. However, there are significant challenges here that plays a big role in wine fermentation specifically. The absence of oxygen is one of the major issues, is that the yeast cannot rescue its plasma membrane effectively in the absence of oxygen. We also don't really understand how the cell senses ethanol in the environment, how it actually goes and responds to that, how that sensing mechanism works. There are a couple of key regulators that have been identified or that are being investigated, but we don't really know a lot more than this about the ethanol response, the ethanol toxicity response. So if we go and we look at nutrient deprivation. Now, I know Dr. Carl Kaiser spoke to you guys about nitrogen already, so I'm not going to rehash all of that again. A couple of things that I want to point out is that during a fermentation, nitrogen does become limited and depleted. What we must take note of, and not many people are aware of this, is some of the hexose transporters, or the proteins, in other words, that is needed for the uptake of sugars for fermentation to proceed. Some of those hexose transporters are turned over in a way that's dependent on nitrogen. If there's not sufficient nitrogen around, these hexose transporters would be degraded by the yeast itself. So consequently, it can't import these sugars, and this will lead to stuck or sluggish fermentations. We know that in the absence of nitrogen, sulfur-containing off-flavors are being produced, like hydrogen sulfide and the mercaptans, so we're not going to go into that in a lot of detail. There's also, in the absence of these of nitrogen, we see the production of higher levels of alcohols and isoacids that give you the off flavors associated with those compounds. And it is also, and this is a strain-dependent phenotype, there's also a decrease in the production of esters, of some esters, as the ethanol, as the nitrogen concentration decreases. So these are all issues that have a significant impact on wine quality at the end of the day. There are two things that we have to take note of when we speak about nutrients. Some of the growth factors or vitamins that are within the juice can become limiting, and they are also associated with off-flavor production, especially with acetic acid and hydrogen sulfide. They've been linked to that already. However, most wine juices that actually come into the winery have sufficient amounts of these vitamins around. And lastly, but not least, lipids can become limiting within a fermentation. We know that as the ethanol concentration increases, the yeast needs to restructure its membrane. In the absence of lipids, that's in the fermentation itself, the yeast will struggle more. So you need to have things like ergosterol and some lipids around for the yeast to actually use. So this can also lead to a sluggish or a stuck fermentation. Okay. So why do we care about these fermentation stresses then? Well, inefficient fermentations, as we've said before, all of these inefficient fermentations can lead to a delay in sugar uptake and consequently in the metabolism of the yeast, and this can affect product quality. So we've spoken about off-flavor production that can increase, and we know what the impact of that is on the bottom line of wines that are produced in these conditions. However, because these fermentations are sluggish, we also open the door for spoilage organisms to develop and to grow. So that's the next layer of contamination or infection that we have to deal with potentially when we run into these problems. So this is a significant detracting from or negative effect of inefficient fermentations. Secondly, dealing with inefficient or sluggish fermentation is a big burden on the winery itself. The winery operations suffer. We have to move wines through fermentation vessels to actually free them up for the next lot that's coming in. So there are significant operational issues that are at play here due to these delayed fermentations, and this decreases or delays the product production. All of this combined leads to losses for our industry. So if we can understand how wine yeast actually respond to these fermentation stresses in more detail, we'll get to a point where we can actually devise strategies to overcome these specific stresses. So if we shift gears a little bit and we move into the yeast and look at some of its molecular biology in a little bit more detail, Wine yeast are really products of domestication. I mean, it's estimated that 6,000 years before BC, we actually winemaking started. So there's been thousands of years in which these yeasts have been exposed to winemaking conditions. So consequently, they've had time to adapt to some of these stresses already, yet they still struggle today. So there has been a significant adaptation that has taken place in these yeasts. If we look at the genomes of these wine yeasts, in other words, at the DNA content of these wine yeasts in a little bit more detail, we see that they are generally diploid. In other words, they've got 2 copies of each gene, or they can be aneuploid, where they either have 2 + 1 or 2 - 1 copies of a specific allele or a specific gene. They are more often than not heterozygous, which means that the 2 copies of these alleles aren't identical. So there can be point mutations between these different alleles. There are, however, some of these strains that are homozygous, which means that the 2 alleles are identical. Now, for the work that we do in the lab, it's much better for us to actually work with homozygous strains, because we can actually go and sporulate them and work with them in a more stable environment. This is not the case when you work with heterozygous organisms. It's a much bigger challenge. Okay, so at least there are strains that was identified by Richard Gardner's group in New Zealand that we can actually work with that are homozygous. We have known for a couple of years, since mid-90s, we've known what the DNA sequence of lab yeast is. So the S288C lab yeast has been sequenced, and we know exactly where which genes are and what they encode. As of yesterday morning, there were still 15% of those genes that we do not know a function of. So we do not know whether or not these genes, where they play a role within the life of Saccharomyces. What a couple of research groups did recently, a group in the Australian Wine Research Institute under Saki Pretorius went and sequenced N96, and Sylvie Decuis' group in INRA in France went to sequence EEC 1118. And they analyzed those sequences and compared them to the known sequence of S288C. What they found is that more than 98% homology exists between the 2 sequences, between lab and industrial yeast, which is great news for us, because at least we know that the genes that we're working with in lab yeast are fairly similar to the ones in industrial yeast. However, and here's a caveat, there are more than 60,000 polymorphisms between these strains. In other words, there are point mutations, insertion or deletion mutations that exist between these, that's present within the industrial yeast, that's not present within the lab yeast. So this is more than likely how the adaptation process took place. How the yeast got accustomed to growing in fermentation conditions and consequently acquired the ability to do fermentations a little bit more efficiently. What is also interesting is that when they compared these sequences of the industrial yeast to that of the lab yeast, they found that there are approximately 37, it varies between the 2 groups, there are approximately 37 new genes that have never been found before in Saccharomyces or in Saccharomyces and its cousins, in the sensu stricto as we know them. So some of these genes are actually associated with other organisms that are also found within the wine or the juice environment. Like Collaborators strains, for example, some of those genes are found in other organisms, which opens the door to the discussion whether or not this would be the first example of a eukaryotic microorganism that has actually taken up genes through lateral gene transfer from another organism, which is fairly interesting because it's never been seen before. But nonetheless, there are genes found in our industrial yeasts that we work with that come from other species, that is not from Saccharomyces specifically, which was a phenomenal finding. Keeping that in mind, we want to understand how the yeast actually responds to fermentations and to fermentation stresses. So how can we measure this and do this at a level that we can actually visualize massive responses all at once? So we can go and measure the amount of RNA or what specific kinds of RNA are present within a fermentation, within yeast at a specific time point. And this will give us an indication of what genes are either activated or repressed. We can also go and actually measure or determine what proteins are present or absent at a specific time point and see all the proteins that are there or are not there, and consequently get an idea of what proteins are functional. And at the end of the day, we can go and determine what metabolites are being produced by the yeast and secreted into the environment, for example, and thereby try to pool all this information and to get an idea of what genes and what proteins are needed for which metabolic responses during fermentation stresses. So this is all things that we can do at a systematic scale, at a large scale. So we started doing this kind of experiments, and we started with doing a transcriptomic experiment. In other words, looking at how gene expression changes during the course of a fermentation. Just because of all this, it's the cheapest to do at that stage in any way. So we started with an experiment that was also done by myself and a previous student from CCOVI, Virginia Marks. And we started with fermenting Riesling grape juice. We had sufficient amount of sugars and nitrogen around and had a good pH. And we inoculated it with an industrial wine yeast. It was Vin 13 at that stage. And we fermented it at 20 degrees Celsius. During the fermentation process, we took samples when 0.5%, 2, 3.5%, 7, and 10% ethanol was produced. So now we know we've got these specific RNA samples, or samples that are collected. We extracted RNAs from these specific samples and then did affymetrix arrays to determine which genes were actually expressed or not. What's the nice thing about affymetrix arrays is it gives you an absolute value, not a ratio between two gene expression points. So because of this, it makes processing the data a little bit easier at the end of the day. So what did we find? So there are 2,550 genes that change more than twofold, which is great, but it's extremely daunting. What are you going to do with 2,550 genes, and how are you going to analyze that data? Right, so this was phenomenal for about a day, and then the reality set in. How are we going to make sense of all of this? So we said, okay, well, let's see if we can determine what the expression profiles of these specific genes were. So if we do a supervised seeding clustering analysis, run an algorithm with this data, and we say, okay, well, we want to know the magnitude of gene expression, how much the gene expression changed, and also what the shape of that gene expression was. So if we apply this algorithm to our data set, what do we come up with? So we ended up with 20 different clusters. And each of these clusters, you'll see a solid line that represents no change in gene expression. And then a dashed line on the other side represents either a twofold increase or a twofold decrease in gene expression. So in the first 6 clusters, for example, you can see an enormous increase in gene expression at the final day, or at the final time point. Right? All of these represent more than a 6-fold change. There was just over 1,100 genes that were induced up to cluster 11. Induced more than twofold. There's about 150, I think, genes that had a transient expression. In other words, it started at no change, were upregulated, and then ended up with no change in both of these instances. There were quite a few genes, about 1,800 for those at the back, that did not change gene expression significantly, and then there was about 1,200 genes that were repressed. Most of the genes that were repressed deals with protein synthesis. And we, at this stage, chose to ignore the repressed genes. And right off the bat, that's a dangerous thing to do. We have to go and follow this up. As you can see, there are 2 proteins right here or 2 genes that are downregulated that play an enormous role in fermentation. The one is a hexose kinase, which is obviously important for carbon, and the other one is needed for the uptake of glutamine. So these are genes that we would like to understand. However, we had to start somewhere. And we called the first 6 clusters, we call the fermentation stress response genes. The reason for that is because the fold change in expression is more than twofold, which is different from the rest of the genes that are upregulated. And already we are dealing with about 220 genes. So we said, okay, well, let's go and analyze those genes a little bit better. And we looked at those specific genes individually. And we saw, okay, and we compared that data set with other stress response data sets that were generated with microarrays before. These first 2 are for environmental stress responses, a variety of them. The second 2 were for osmotic stress response, either with sugar or with salt. This is a ethanol spike and nitrogen deprivation. So when we looked at our genes that we found, and this is just an example, in cluster 1 and 2, we found 2 heat shock proteins that were upregulated, and they were known to be upregulated through stress responses before. They significantly increased in transcription, and we see that they were identified in previous stress responses. The same thing with trehalose biosynthesis, and we said that trehalose is synthesized when yeast experience ethanol stress. Same thing happens here. It's been seen in previous experiments already. So these were our positive controls, and we could identify those genes. However, what's interesting is that we found a variety of proteins that have not been previously identified in any of the other stress responses, right? These guys over here. Also, what's interesting, there are a variety of proteins here that deals with nutrient metabolism, like hexose transporters, for example, HXT7, methionine biosynthesis, T3, and then also the uptake of amino acids, DIP5. These are all related to nutrient responses. There are also a couple of genes that have unknown functions that we don't really know what they do. So this opened up a whole new field of investigation. Why are these genes that have no known function yet, why are they upregulated during fermentation? What is their function at the end of the day? So I said, well, let's categorize these genes into functional categories. And as I mentioned before, quite a few proteins, that about 18% of the proteins of the genes are related to transport in some way. There are quite a few proteins that deal with carbohydrate metabolism, about 8%, and there's around the stress response genes, around 11% of them were identified. However, there was about 23%, about 60 genes that didn't have any known function that we wanted to investigate and say, okay, well, why are these genes, what are the functions of these unknown genes during the fermentation process? So I'm going to focus on some of these proteins for the rest of this talk. We've already identified one of these genes as a transcription factor that was previously unknown to play a role in carbohydrate metabolism. We've renamed this to nutrient stress response factor, but I'm not going to talk about that today. I'm going to talk about these three proteins over here. We know that HXT7 is the hexose transporter, and then there are two proteins, the VID proteins or genes, that I want to speak about a little bit. So if we look at the hexose transporter gene family, there are 17 hexose transporters that we know of in lab yeast. There's some debate about that in industrial yeast. A lot of literature says that there are some of these hexose transporters that were actually lost during the domestication process, but we're still investigating that. So in lab yeast, we have 17 of these hexose transporters, and they have different affinities. They play a role in transporting glucose and fructose into the cell for growth. So they function in fermentation because glucose and fructose are the main sugars thought to be present in equimolar amounts in grape juice. So there are hexose transporters with different affinities. We have the low affinity hexose transporters, HXT1 and 3, for example, that function when there is an abundance of glucose around or fructose around in the environment. There are ones that have moderate affinities like the HXT5, and then ones that have high affinities that function when the cell experiences low levels of fructose or glucose in the environment. I highlight these two because we're going to revisit them later on. Interestingly, these hexose transporters have different affinities for glucose and fructose. They like taking up glucose faster than fructose. So at the end of the day, when you have a stuck fermentation, what's left is fructose rather than glucose. And of the two, fructose is the sweetest. So that's why you don't like selling stuck fermentation wines, because you can taste this fructose that's left. What's actually important is that this HXT7 functions at the end of the fermentation, so we take care of fructose, and HXT3 is also thought to play a role to take up most of the fructose during the fermentation. So these are important proteins. The way that the cell actually regulates these HXTs is to regulate its transcription. So as the glucose concentrations or sugar concentration changes, it'll change the way in which it expresses these genes. And it can also control its degradation. So it can actually go and target specific hexose transporters for degradation. So if we look at how these hexose transporters are expressed during the fermentation process, and we look at our data set, we see most of these hexose transporters are activated in cluster 11. So where they are initially upregulated during fermentation, but then downregulated towards the end, right? Which implies that there would be less protein at the end of the fermentation. That's a dangerous assumption, because we assume because there's less gene transcription, there would be less protein. And those two aren't necessarily linked, as literature will show us. Nonetheless, most of these hexose transporters are activated during fermentation, their transcription at least. If we now look at protein turnover, and here you've got to bear with me. We find an excess transporter within the plasma membrane when, for example, there's a certain amount of glucose or fructose around in the environment. It's brought into the cell and it's converted into ethanol. Ethanol is secreted from the cell. So we find it within the fermentation itself. So what happens now if we actually go and run into a situation where there's no more glucose around, or low concentrations of glucose? And this would, for example, be HXT3 that needs a high concentration of glucose around. It is post-translationally modified in that other proteins are added onto it. These proteins are called ubiquitin. So they're added onto this excess transporter on the plasma membrane, and consequently they are internalized into endocytic vesicles and consequently removed from the plasma membrane. So now these excess transporters can't perform their function anymore because they're not in the plasma membrane and consequently can't move anything into the cell. Endocytic vesicles make their way into the vacuole at the end of the day, which is where most of the degradation of plasma membrane proteins take place, and consequently we lose that protein. It has degraded, it's got no more function, it's not a reversible process. The same thing happens with enzymes that function in specific nutrient conditions. So, for example, in the production of a certain metabolite that's a flavor compound, once that enzyme is not needed anymore, it's tagged also by ubiquitination and it's targeted to proteasomes, which are enzyme clusters or a degradation site within the cell itself, and it's degraded. Also a non-reversible process. So this takes us back to this graph, and more specifically to these VID genes. These VID genes, there are a family of VID genes, there are about 13 of them. And they've all been identified in that they play a role in the degradation of proteins in response to a nutrient shift. In other words, they can actually degrade or is responsible for the degradation of a specific enzyme when that enzyme isn't needed anymore. And it's been studied extensively with only one enzyme. And this enzyme is FPPase, which is needed to produce glucose at the end of the day when the cells is grown in the presence of ethanol. Right, so this is a reaction that the FPPase would do on ethanol as a carbon source. However, as soon as you change the carbon source to glucose, this reaction is not needed anymore. FPPase is ubiquitinated and targeted to either the proteasome or the vacuole for degradation. Right? And the VID genes is needed for this entire process. Okay. But the immediate caveat here is we're looking at in the conditions of glucose where these VIDs are functional. This is exactly the opposite of how fermentation runs. Fermentation starts with a high concentration of glucose and moves into ethanol. And these genes were shown to function in the other conditions, in the opposite conditions. So what interested us in the VID complex to start off with is that it was shown, one of its components was shown to interact with HXT7, which I knew didn't function when there was a high concentration of glucose around. It functioned actually in the opposite conditions. So my question was, okay, well, how does this work? How do these VID genes actually interact and play a role somehow through HXT7? So we did some fermentations, trial fermentations, just with lab yeasts of some of these VID mutants, and we saw that some of them actually fermented glucose faster than the wild type would, indicating that there is definitely a link between carbon metabolism and the ability to use glucose and these VID genes. So we went on a quest to actually try and understand that a little bit more. We queried the data set that we had for the VID genes. So the first 13 proteins over here are all part of this VID or GID gene family. And all of them are activated somehow during the fermentation process. Find them in the first 11 clusters. So what we wanted to do is to see, well, how are these genes linked to HXT7. So the question that we had to ask is, okay, we have to develop an assay of some sort, how we can actually see or visualize or interrogate for HXT7 when we are studying these VID genes. So we said, okay, let's do it the easy way and see if we can fuse our HXT7 gene to a protein tag or to a molecule that we can detect using a fluorescence microscope, for example. So if you can imagine that this would be a chromosome on which we would find the HXT7 gene. It has a promoter region that drives its transcription and a terminator region that will stop the transcription. What we can now do is we can go and make a tagging cassette through PCR in the lab that has a site that's homologous to the 3' end of the gene. It has a sequence that's homologous to the terminator sequence of the gene itself, and it has this green fluorescent protein or GFP in between these two sequences. We can consequently transform this into our yeast of interest and thereby generate a chimeric gene in which H67 is fused to GFP itself. When this protein is therefore, this chimeric protein is consequently transcribed off the native promoter of H67 itself, we should get a protein that actually has a GFP tag to it and that would localize to the plasma membrane, and we can see this with a microscope. So the next question we had is, okay, if we go and tag H67, for example, in some of our VID mutants, can we see that there's some implications for its subcellular localization or turnover? So we said, okay, let's work with VID28 first, because that's the protein that we studied at that stage in the lab. We know that through nitrogen starvation, H67 gets turned over. So if we take a wild type yeast strain in which H67 is tagged with GFP, you can see this plasma membrane localization of H67 when there's still sufficient nitrogen around. In these conditions, there are low concentrations of glucose present in the environment. As we starve the cells for nitrogen, we see that this H67 is depleted from the plasma membrane and ultimately becomes turned over where you can't visualize it anymore. This was supported by other experiments as well, but I'm just showing you the bottom line here. When we knock out VID28, we see that this internalization that we saw in the wild type is delayed in the mutant. After 24 hours of starvation, you can see some H67 in the plasma membrane at the end of 24 hours. This means that in this VID28 mutant strain, the protein is potentially still functional at the end of a 24-hour nitrogen starvation because it's still in the plasma membrane, which has massive implications for addressing things like, for example, stuck fermentations, where you could have a exose transporter that's present in the plasma membrane longer than it should be and can consequently help with finishing off fermentations. So we said, okay, well, let's go ahead and knock out another gene, because we know that there are two components that are really important in this or this VID gene family. So we knocked out VID28 and VID30, and we see there's even a further stabilization in the plasma membrane after 24 hours of nitrogen starvation. Right, so the implication here is that these two proteins actually have redundant functions in keeping in the turnover of H67 at the end of the day. So let's go back to our data set. We identified VID27 and VID24 as proteins that are needed or that were transcribed actively in the fermentation stress response. So we did the same experiment in VID27 and VID24 knockout mutants, and we see in both instances that there are still exose transporter present within the plasma membrane after 24 hours of starvation. This is not so apparent, not so clear in the VID24 mutant. But it's very significantly, it's very clear in the VID27 mutant where you still have this in the plasma membrane. Okay, so that's H67. So we know that these VID genes are all play a role in the turnover of H67, and in the absence that this exose transporter is stabilized in the plasma membrane. So we said, okay, VID27 is known to play, H67 is known to play a role at the end of a fermentation. What about H63? This is the low affinity exose transporter, the one that actually functions when there's abundant sugars around. The interesting thing about H67 is that it's been linked to the uptake of fructose as well, which is the problem child when it comes to fermentations, because it's left at the end of a fermentation. It's the last sugar to be taken up. So we wanted to know whether or not H63 turnover is dependent on our proteins as well. So we know that if you grow glucose or cells in the presence of glucose and then shift it to ethanol, that the transcription of H63 goes down, and then this protein is now consequently turned over as well. As you can see that in the wild type strain where H63 is now tagged with GFP, you can see the presence of H63 in the plasma membrane. This is expressed at lower levels than H67, so it's not as clear. But over a period of time, you actually lose this plasma membrane localization up to a point where you can see hardly anything after 6 hours of a shift to ethanol as a carbon source. When we look at our double mutant, we knock out VID28 and VID30, we see that after 6 hours, it's still present within the plasma membrane. So this is a different set of conditions, remember. So this is a condition where you go from glucose to ethanol, which is opposite to what the researchers saw with FPPAs when they analyzed these VID mutants. And it's also not nitrogen starvation. So this tells us that the turnover of H60 of these exose transporters that are dependent on both nitrogen source and on carbon conditions are all dependent on the VID genes, which implicates this complex to function in a variety of different conditions when it comes to the turnover of proteins. And these all seem to have an implication of internalizing these excess transporters and consequently removing them from the plasma membrane and not having the yeast be able to do fermentations. So this is a really exciting avenue for us to research the potential of overcoming stuck fermentations at the end of the day. I have 10 more minutes. I hoped I would. I want to switch gears with you a little bit and talk about a different protein that was actually up to about a year ago had no known function. This protein is called, we called it ethanol tolerance protein 1, and this protein was identified in a large-scale experiment. It was identified to interact with one of the components of the vid gene family, specifically MO1. So these two proteins were shown to actually interact with each other. What we thought is, okay, it's interesting for us because this association does exist. So we started as a side project. I put an undergraduate research project student on this project, and we wanted to know whether or not ETP1 has any specific function that's even vaguely similar to what the vid genes have. So we knew at this stage, through all the microarray experiments that were done, we knew that ETP1 was highly induced by ethanol. Its transcription went up significantly. So we thought, okay, well, let's see, does it have a function? Right, so we said, okay, well, let's get a mutant. So we had a wild type strain, we had a couple of mutants, and this is MO1 itself that interacts with ETP1, and there's a third protein that also interacts with ETP1. So we said, okay, let's do spot assays, just a typical standard lab spot assay. We spotted equal amounts of each of these strains in a dilution. This is 10 to the minus 1, minus 2, and minus 3. We spotted this to a rich media in the absence of ethanol, and all the strains seemed to grow happily. As soon as we actually added ethanol to high concentrations, so it actually gets the yeast to struggle with these conditions, we see that all of them grow similarly except ETP1. It seems to grow a little bit weaker than the rest of them. So we said, okay, well, there's something here that obviously plays a role in tolerating ethanol in some way, shape, or form. So we said, well, we know that the heat shock proteins are actually transcribed and activated in a mechanism that's dependent on ethanol. So let's see whether or not some of these heat shock proteins are transcribed in a manner that's dependent on ETP1, potentially, because it struggles to grow in the presence of ethanol stress. So if we look at our control conditions where YPD served as the rich media served as a growth medium, we had our wild type strain and now ETP1 mutant, and we couldn't see any expression of either HSP26 or 12. We used actin as an internal standard as a loading control for these experiments. If we overexpose this blot, we saw these bands come up at the end of the day. So there was a basal level of transcriptional activation of these two genes. When we converted that into a graph using ACT1 as a standard, and we equated each of these wild type levels to 100%, we saw that for HSP26 there was about a 22% decrease in transcription in the ETP1 mutant, but there was almost no change in transcription of HSP12. However, the interesting bit is when we actually stress these cells by adding ethanol to the growth medium, we see that there's a significant decrease in transcription of these HSP genes when analyzed in the ETP1 mutant compared to the wild type. When we actually did the same experiment by looking at a graph to represent this data, we see that there's more than a 60% loss of transcription of either of these two HSPs in the absence of ETP1. So this is a prime candidate then for why these yeast cells will struggle to grow in the presence of ethanol, because these chaperone proteins aren't there to protect the proteins from unfolding and to help them refold. But we wanted to ask the question whether or not ETP1 is related somehow to the vid genes. So we said, okay, let's look at HSP3 turnover. We know that the vids are needed for the turnover of HSP in response to ethanol stress, or in response to an ethanol shift from glucose to ethanol. So again, in the wild type cell, we tagged HSP3 with GFP, and we can see turnover that took place, and this is significantly delayed in the ETP1. So here we've got two phenotypes of the ETP1 mutant that has a bearing on fermentation. The one is it protects, or it's needed for ethanol tolerance, and the other one is that it actually delays turnover of HSP3. So because the mutant is more sensitive to ethanol tolerance, it's not really a big plus for us, because we thought, okay, well, we don't know whether or not when we do fermentations with this mutant is going to croak over the fermentation process. And the other thing that we didn't know is whether or not this resistance to turnover HXT3 would have a stronger phenotype than ethanol tolerance. In other words, would the strain still be able to ferment and consequently give us a handle on a better fermenter at the end of the day? So we went and we did a fermentation assay in which we took M2, which is a homozygous diploid of industrial yeast, and we generated the mutants in which we had a heterozygous mutant and a homozygous knockout of ETP1, and we did a normal Chardonnay fermentation. And we measured weight loss, in which we weighed the fermentation vessels during the course of the fermentation, and this is a daily weight loss graph, not a cumulative one, in which we measured basically the liberation of carbon dioxide. And as you can see here with a wild type, you see a normal profile with a massive amount of carbon dioxide lost early in the fermentation and then it peters out towards 2 weeks. The heterozygous diploid shows us a very similar profile. There's a huge error here, a standard deviation error here that we have to sort out with some follow-up experiments. But what was interesting is that the homozygous diploid for ETP1 couldn't ferment, which indicates to us that ETP1 is really critical for the yeast cell to actually perform fermentation efficiently. Now, we do not know at this stage why this is the case. We suspect that it's because it cannot transcribe or it has a lack of transcription of the heat shock proteins. However, we have not done metabolite analysis or any kind of gene transcription or proteomic analysis on this strain, which would obviously be the next step to see why this protein, ETP1, is so important for the fermentation process. So, our conclusions is, well, we used functional genomics to investigate vinification, and we identified the fermentation stress response. This is an integrated response in which we see fermentation stresses like ethanol production and also like metabolite, the nitrogen starvation and nutrient deprivation characteristics all in one stress response. There were 60 of these genes that are uncharacterized without any known function. We've identified 2 VIDs that are actually part of these 60 genes, and we've investigated some of the other VID proteins as well, and see that they play a role in nutrient-dependent degradation of these exose transporters, which has enormous bearing on the fermentation process. We've also identified a new gene that plays a role in ethanol tolerance. We do not know how these things function other than what I've explained to you today. Got some ideas that I'm not sharing with you, but I'll keep that a secret. What we want to know next is how do these VIDs and the ETP1 protein, how do they impact wine quality? So we have to go into metabolite analysis and wine quality analysis, which would be the next step. We also want to do proteomic analysis of these specific mutants during the fermentation process. There has some work being done on the proteomic adaptation during fermentation, so looking at the complement of proteins during fermentation itself. What we want to know is whether or not these 2 sets of genes actually play a role in this proteomic adaptation. Both of these sets of proteins look like they can be part of a family of ubiquitin ligases, which are proteins that are needed for protein turnover. So by doing proteomics, we might be able to identify more targets of the VID proteins and potentially the ETP1 protein as well. And in combination, all this data would help us to actually develop strategies to overcome fermentation-related stresses. The people that did the work, I'm purely a guy that stands up here and tells you what all these people have done. This is members of the lab, past and present. The work that I showed you today was mainly the work of Chris Snowden, who's a senior PhD student in my lab. He's done phenomenally well. And that of Ashley Hillier, who's a new master student in my lab, did 2 years of research, 2 semesters of research project work. She worked on ETP1. As well as Stephanie Hughes and Peter Polichuk. They are students that worked on the ETP1 story. We collaborate very closely with Debbie here at Brock and Barry Shelp at the University of Guelph that does some metabolite analysis for us. He's in the process of actually looking at some of our mutants as we speak. On the gene expression, on the FSR experiment, we collaborated with Henny van Vuren and Vivian Measday out at UBC, and I collaborate constantly with Terence van Rooyen at the Niagara College Teaching Winery. where we do some of our fermentation trials. The people that pay the bills are these guys, and I thank you for your attention.
Thank you, George. We've got time for some questions. I have a first question.
Sure.
In your research, you looked at knocking out ETP1
Yes.
and saw the impact. Have you looked at overexpressing it?
That's the next step.
Is that the next
Yeah,
step?
so that's absolutely correct. Obviously, we now know that the yeast's ability to respond to ethanol stress is just diminished in the absence of ETP1. Now we've got to do the copy number experiment. We see that even if one copy is present, that it can have wild type levels of fermentation, indicating that this gene has a really strong function within the cell itself. We're looking at increasing the copy number just by a single copy and then also overexpression to see whether or not we can potentially recover. The flip side, what we're worried about, is whether or not we're going to disrupt stoichiometry with the turnover of excess transporters. But you don't know. You don't know until you've done the experiment. So yeah, doing the overexpression experiment is next in line.
Although even if you did that experiment and didn't get the result maybe you were anticipating, it also may speak to the stoichiometry of,
Yes.
of how that complex
Yes.
is actually working.
Absolutely.
If it's part of the turnover.
Yeah, absolutely.
Also, just a further follow-up, what I see the huge value in the work that you're doing, you're really starting to narrow down some target genes and their protein function that we can use in the future to be able to select yeast that have this particular profile or that to overcome these various fermentation stresses.
Also the targets. We're coming at this is something that people get confused about quite often, is that we're coming at this from a mechanistic point of view, is that we want to understand how it works. But the protein targets of this process is very important. For example, HXT3, there are alleles that have been identified recently in the literature in which HXT3 is not turned over as it should be. And they've identified what that specific allele was. If it wasn't for the fact that we understand the mechanism, we wouldn't have known what to look for. And consequently, the allele was identified, which could help us with doing more efficient fermentations in the future. So not only the mechanistic, but also the protein targets itself. It's very important to understand what they are. Yeah.
Are there any other questions? On group today? Any questions online? Well, with that, George, I'd like to present you a small token of our appreciation for your time today and presenting such a stellar piece of work. Just fantastic research. I'm really excited to see how this is going to progress.
Well, this is not necessary, but thank you.
You're very welcome.